Good morning, everybody. Myself, Professor Ramchandra, Head of Department of Environmental Microbiology, Babasaheb Bhimrao Ambedkar Central University, Lucknow. In case of the bacterial nutrition, if you will see their mode of the nutrition, they are actually classified into autotrophic and heterotrophic. Under the autotrophic, there is a chemoautotrophic, photoautotrophic, and in case of the heterotrophic, there is a saprophytic, parasitic, symbiotic. And if you will further divide the photoautotrophic bacteria, there is a major group is green sulfur bacteria and purple sulfur bacteria. In case of the chemoautotrophic bacteria, there is a lot of group of the bacteria. They are the sulfur bacteria, hydrogen bacteria, iron bacteria, methane bacteria, nitrifying bacteria, and carbon bacteria. So this is the in brief regarding their nutritional properties of bacteria. The bacterial cell wall divide independently as a asexual mode of the reproduction, and that is known as budding. But two cells may fuse together to exchange their genetic material for reproduction by three means mode, that is known as transformation, conjugation, and transduction. If bacteria accumulate the genetic material from their media, that is known as transformation, and if there is a exchange of genetic material from conjugation tube, that is known as a conjugation mode of their reproduction. When there is a phase mediated transfer of genetic material, this is known as the transduction. Relevance of bacteria in the environment. What is their importance? They actually maintain the biogeochemical cycle, they help in biodegradation, and the bacteria play a central role in the ecosystem to maintain the cycle of the element, and a lot of the activity of the environment is maintained by the bacteria. Used for making medicine also, there are lot of bacteria produce antibiotics. They are also useful for development and increase productivity in agricultural crops, and they are also very important for various industrial application, because in industry the fermentation process or they can produce many useful product. They regulate the digestive system of the animal, including the humans. So in this way, broadly, if you will see the role of the bacteria, they are very important to maintaining the environment and human life also. Another group of the bacteria, which is a very important, what I told you regarding the bacteria, that is the general property. But if you look further, the detail of the bacteria, there is very one unique group of the bacteria that is known as archaebacteria. Archaea are the prokaryotic unicellular organism. As per the literature meaning, archae means the ancient, and this is the Greek word. Archae is the beginning. Archae means the beginning. Archaea were originally described as extremophiles living in the extreme environment such as springs, hot springs, the uh, thermoproteota being the most common form of the life in the ocean, dominating ecosystem below 150 meter in the depth, and these organisms are also common in soil and play a vital role in ammonia oxidation. This is the most diversified group. It is estimated that around 5 nonillion, means 5 into 10 power 30 cells, accounting for at least half of the biomass on the earth. Therefore, In 1990, microbiologists proposed to divide a separate group of the archaea from the bacteria because the archaea differ from the bacteria in both their genetic and biochemistry. For example, archaean membranes are made up of ether-linked isoprenoid side chain, while in case of bacterial system there is a ester linked hydrocarbon in membrane lipid. So there is universal lack of muramic acid also as a constituent of the cell wall. Peptidoglycan in contrast in its universal presence among the eubacteria. So, in case of the archaebacteria, muramic acid is absent, while that is present in case of the other bacteria. And similarly, they have also their membrane unique property of the ether-linked isoprenoid side chain. These two property is the unique property of the archaebacteria which differs from other bacteria. So, in this way, if you will see the structure of their cell wall composition, the glycerol diphytonyl chains and glycerol phosphate, glycerol and diphytonyl chains is present, which is the characteristic feature of the archaebacteria. This is the ether lipid membrane uh, linkage. If you look their group of the archaebacteria, they are actually broadly divided into three groups. They are as a methanogens, thermoacidophiles, and halophiles. Among them, methanogens is very common. We are aware with the methanogens in anaerobic environment. So they are strict anaerobes. They occur generally in marshy area or in wetland ecosystem. The organic rich condition fermentative bacteria produce formic acid, acetic acid, CO2, hydrogen, which is subsequently converted to the methane by methanogens. So, methanogen is important in anaerobic environment for methane production. Since methane is a gas that is very less soluble in water, it escapes from anaerobic environment if it is not released immediately outside from the water, the another bacteria become active which consume the methane, that is known as methanotrophs, usually act at the interface between anaerobic and aerobic condition as shown in the figure. You can see if there is a methanogens at the bottom, looking the blue color, that is the water suppose, and the methanogens is producing the methane and it is coming out in the environment. If it is delayed, if it is delayed, then another group of the bacteria that is known as methanotrophs and it comes and eat to the methane. So this makes the methane oxidation and simultaneously there is also release of the CO2. This picture also is showing the process of the methane formation and role of the methanogens in the environment. If we will see in the wetland ecosystem or marshy land or submerged condition, there is a organic rich condition and organic compound. Suppose through fermentation they produce formic acid, acetic acid, or CO2 and hydrogen. This process is known as acidogenesis, and subsequently with the help of the methanogens they produce the methane, and the methanotrophs, if they are not releasing very frequently, that provides the environment for the methanotrophs and they actually utilize the methane gas and they are aerobic. So, and this there is a, you can see the right side of the picture, there is a aerobic interface and anaerobic. So methanogens actually grow in the anaerobic condition. In interface condition there is a methanotraphs, and aerobic condition the methane comes out. So, this way methanogenic archaea population play an indispensable role in anaerobic wastewater treatment. Also, for example, methanobacterium and methanococcus is a good example for this group of the bacteria. In anaerobic environment of sewage digester or sewage treatment and some sanitary landfill, methanogenesis occurs at rate that make collections and compression of this principal component of natural gas economically feasible. But in some anaerobic environment in which methanogenesis is not quantitatively important, a comparable ecological role is played by sulfur reducing eubacteria. So, the factor that determines which of the two groups will predominate is not well understood. An affinity of uptake system for acetate and hydrogen is supposed to be a key factor regarding the preferential growth of the sulfur reducing bacteria and methanogens. That is, there is a higher in case of the sulfur reducing bacteria for the affinity of the towards the acetate than in methanogens, thought to contribute a key role for their domination. If you will see their correlation in the environment regarding the acidogens and methanogens, you can see the CO2 is converted with the help of the hydrogen into acetate. Simultaneously, there is a CO2 and acetate convert into the methane by the methanogen, and simultaneously the hydrogen is also taken by the sulfur reducing bacteria with the help of sulfate into the sulfide. So this sulfur reducing bacteria and methanogens survive into the same environment with the preferential their substrate by maintaining the environment or situation. If so, this the process is very specialized and is specific. If any parameter is deviating, their equation or balance is disturbed. So there will be either domination of the SRB or sulfur reducing bacteria, or there will be a major growth or domination of the methanogens. So the balance of SRB and methanogens in the nature is very, very important and specialized. It is well known that under anaerobic condition, mineralization of complex organic matter requires cooperation between at least three trophic groups. The first step in the degradation of organic matter is known as hydrolysis of complex molecule into their oligomers or monomers. Means at the first step there is a hydrolysis, means conversion of the complex compound into simpler form, and this step is followed by fermentation involving the degradation of this substrate to reduced organic compounds such as short chain fatty acid and alcohol. In sulfate, while in case of sulfate rich sediment, sulfate reducing bacteria or SRB can use the product of primary fermentations and oxidize them to CO2. So, as I pointed out in my earlier line, that the balance between SRB and methanogens is very, very important depending upon their concentration of the sulfur or presence of the acetate. However, in sulfate depleted methanogenic sediment, starch and fatty acid and alcohol are converted by secondary fermenter to acetate, are formed, are hydrogen and CO2, which are subsequently utilized by methanogenic bacteria to produce the methane gas. In fact, acetate is a key intermediate in marine sediment also, as it is one of the major end product of fermentation and serve as a primary substrate for several terminal electron accepting process. Like sulfate reduction and methanogenesis, there are two possible processes for methanogens to produce methane from acetate. In the first process, acetate is cleaved to methane and CO2. This process is called as acetoclastic, acetoclastic and methanogenesis, and it is an energy yielding reaction under standard condition. The second process, that is known as a syntropic acetate oxidation. In case of the syntropic acetate oxidation is a two-step process. If the in case of the first step, acetate is oxidized to CO2 by an acetoclastic microorganism with generation of reducing equivalent often as the hydrogen, and this step is endergonic and requires a hydrogen nootropic microorganism for the consumption of the produced hydrogen. In the second step, hydrogen nootropic methanogens scavenge that hydrogen and the overall reaction become thermodynamically favorable. Hydrogenotropic substrate reducers can also be involved in second step, and in case of the SRB or sulfur reducing bacteria as a partner organism, the overall reaction is the same as if sulfate reducer would oxidize the acetate completely without syntropic partner. Almost all acetate in sulfate zone is converted to CO2, not to the methane, suggesting the predominance of acetoclastic sulfate reduction. So, the role of their substrate is also very important to determining the growth of the SRB and the methanogens and this prevalence of the reaction. So thermodynamic mechanism to explain the biogeochemical zonation in marine sediment in the presence of acetate are unclear so far. So thank you very much. I will be meeting in the next class.